So to briefly summarize, glycolysis can be divided into two parts: the preparative phase, shown here in white, and the energy-producing phase, shown here in gray. The net result of glucose oxidation through glycolysis is 2 ATP, 2 NADH, and 2 pyruvate. So the process of glycolysis starts with the phosphorylation of glucose by hexokinase or glucose kinase. And this phosphorylation to glucose 6-phosphate traps the glucose within the cell. From here, it can undergo an isomerization to fructose 6-phosphate and further phosphorylation to fructose 1,6-bisphosphate. From here, the fructose 1,6-phosphate is cleaved by aldolase B into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These 3 carbon units of glyceraldehyde 3-phosphate are ultimately used to produce pyruvate, and under aerobic conditions, the pyruvate will enter into the mitochondria and be oxidized to acetyl-CoA, which will enter into the TCA cycle. Conversely, when oxygen is limited or energy demands exceed oxygen demands for ATP, the cell will rely on anaerobic glycolysis. So glycolysis has 3 regulatory enzymes, and these are also our irreversible steps when we talk about gluconeogenesis. Those enzymes are glucokinase or hexokinase, phosphofructokinase 1, and pyruvate kinase. So let's start with glucokinase or hexokinase. This kinase will trap glucose in the cell by phosphorylating it to glucose 6-phosphate. It's important to note that hexokinase is the enzyme responsible for this phosphorylation in most tissues, whereas glucokinase is found in the liver and the beta cells of the pancreas. This is relevant as the kinetics of these enzymes, hexokinase and glucokinase, differ significantly. So if we look at the kinetic profiles of these 2 enzymes, you can see they perform very differently. Glucokinase in the liver has a higher Km for glucose, or a lower affinity when compared to hexokinase. That means in the liver, this enzyme will phosphorylate glucose only when glucose concentrations are high, such as in the fed state. Hexokinase, on the other hand, has a low Km or a very high affinity for glucose. This enzyme becomes rapidly saturated by glucose and only takes up glucose into the cell over a very small range of concentrations. So our next enzyme, phosphofructokinase 1, is responsible for phosphorylating fructose 6-phosphate to fructose 1,6-bisphosphate. So the regulation of phosphofructokinase 1 is primarily through allosteric regulation by AMP and fructose 2,6-bisphosphate. High levels of AMP would indicate a lack of energy within the cell, and this would increase flux through glycolysis by enhancing the activity of our enzyme, phosphofructokinase 1. Conversely, phosphofructokinase 1, or PFK as you'll hear it referred to, is also inhibited by high levels of citrate and ATP. Levels of these compounds are indicative of high energy states, suggesting there's sufficient oxidation products. Under these conditions, glucose is diverted to storage pathways. Now let's talk for a minute about fructose 2,6-bisphosphate. This is an important regulator of glycolysis, and it's formed by a shunt in the glycolytic pathway. You can see that here. When there's excess fructose 6-phosphate in the cell, the substrate is accepted and phosphorylated by our enzyme phosphofructokinase 2. The compound fructose 2,6-bisphosphate functions as an allosteric activator of phosphofructokinase 1. Additionally, our enzyme phosphofructokinase 2 can be regulated by covalent modification. This enzyme is a bifunctional enzyme and only functions as a kinase when insulin is high and the enzyme is dephosphorylated. We'll see later when we discuss gluconeogenesis under fasted conditions when glucagon is high. This leads to a phosphorylation and inactivation of phosphofructokinase 2. This also leads to an activation of a phosphatase function of the very same enzyme. Our final regulatory enzyme is pyruvate kinase. This converts phosphoenol pyruvate into pyruvate. So pyruvate kinase can be regulated by phosphorylation and by allosteric means. This enzyme is subject to feed-forward activation by fructose 1,6-bisphosphate, which allosterically activates the enzyme, increasing flux through glycolysis. Conversely, as energy levels in the cell increase, high ATP levels will reduce the enzyme activity. Pyruvate kinase can also be regulated by glucagon-mediated phosphorylation. In the fed state, pyruvate kinase is dephosphorylated and active. However, in the fasted state, as glucagon levels elevate, this will induce a cascade of events leading to the phosphorylation and inactivation of this enzyme. So based on what we know about metabolic shifts, which of the following would reduce the activity of the glycolytic pathway? Decreased alanine, increased glucose, increased ATP, or decreased ATP? The correct answer here is increased ATP. We'll see as a common theme that high levels of ATP or NADH is highly regulatory of metabolic pathways, as these are indicative of high energy states, suggesting that we don't need to oxidize any more fuel products, rather we should transition to storing them.